Yes, sir.
Okay, so let's start with a brief summary of the last class. We started with the structure, right? We did the central dogma already. We started with the structure of the DNA. Okay, so who will give me a brief recap of the last class quickly? Miraj, will you do that? Are you there, Miraj?
Yes, sir.
Yes. So would you like to give me a quick recap of the last class? And also to help Madiha.
Okay, sir.
Yeah. Okay.
We
Your voice is breaking, Miraj. You're not audible to me.
So am I audible now?
Yes, you're audible now. Continue. Good.
So we started with DNA structures. We did one, the nitrogenous base, and then how the DNA, how the RNA and DNA are formed. DNA is
Yes.
nucleic acid, and DNA is deoxyribose nucleic acids. Okay.
Yes.
And third, we did the phosphate group. The how the DNA is formed, the structure of the DNA, and then, sir, we did. Sir,
So
after
so
that,
these
you
three
explained to us
these
how
three
all of these make the nucleoid,
yeah, Miraj. So these
make
three
the nucleoid.
components are the components of the DNA. So DNA, in a DNA structure, there are three things: a nitrogenous base, a pentose sugar, which in case of RNA is called ribose sugar, and in case of DNA is called deoxyribose sugar. And the third thing is a a phosphate group. Phosphate. There has to be a T here. Yes, phosphate group. And this is how these three are arranged, right? This is the phosphate group at the 5' end, a sugar and a nitrogenous base coming, which is projecting inwards from the backbone. So this is the backbone of the DNA made up of sugar and phosphate. And this is the base coming inside. Yes, continue, Miraj.
So, sir, this structure is called the polynucleoid chain.
Polynucleotide.
This uh,
And what is the backbone? So this I already told you. The backbone is made up of phosphate group and sugar. Yes. Now tell me about the characteristics of the DNA, which are on the board. You see this, Miraj? The points on the board? I think he's having some issues.
yes, sir.
Yes. Would you like to continue, Miraj? Or let me go to Wafia. Wafia, are you there? Would you like to continue from here?
Yes, sir.
Yes, sir.
Yeah. Thank you.
So the DNA is acidic in nature and it is negatively charged.
Mhm.
And it was DNA, the acidic nature of the acidic substance was present in DNA was first discovered by Frederick Nietzsche in 1869 and he called it nuclein.
Yes, he did not know that it is DNA. Okay. He knew that something is present in the nucleus and he called it nuclein.
Yeah, he called it nuclein. Then
Yeah.
Rosalind Franklin and Morris Wilkins, they conducted an X-ray and they collected the data, but they couldn't explain the data. Then in 1958, James Watson and Francis Crick, they took the DNA, which was found by Rosalind and Wilkin, and they proposed the double helix
They took the
structure.
data, not the DNA.
Data of the DNA and proposed the double helix structure of DNA.
Yes. And what are the features of this double helix structure?
uh, it is the double helix structure. It is made up of two polynucleotide chains, and the two chains are anti-parallel to each other, and the basis is opposite strands are paired through hydrogen bonding. That is, adenine is double bonded with thymine, and guanine is triple bonded with cytosine. Then two
Yes.
chains or strands are coiled in a right-handed fashion, and the distance between two base pairs at 0.34 nanometers. Okay, then the plane of one base pair stacks over the other in a double helix. This stacking along with the hydrogen bond makes DNA more stable.
Yes.
Then the packaging of DNA, and the distance between two consecutive pairs of base pairs are 0.34 into 10 raised to minus 9 meters, and the and the
Total.
total length and the
Yes. Yes.
total base pairs in a mammalian cell is 6.6 into 10 raised to 9, and total length of DNA in one cell is 2.2 meters, and it is formed in a supercoiling of DNA is happen, and DNA is negatively charged and is packed with the help of positively charged proteins known as histone proteins.
Yes.
Then steps of packing. The naked DNA is formed to nucleosome, then to chromatin, then to chromatin fiber, then to chromatid, that is chromosome.
Okay, and what are these 4 histones, 200 base pair of DNA wrapped linker DNA?
Yeah. The
So
DNA.
DNA is wrapped around this core histone protein, which is made up of 8 smaller proteins. So it's known as histone octamer, right? And this is positively charged. So the DNA that is packed around this octamer is also put kept in place by this H1 histone. Again, all the histones are positively charged. Now, this kind of a structure where a DNA is wrapped around the octamer, there is some DNA in between which is left. Okay. Now, this DNA which is left between two nucleosomes. So this structure of a DNA wrapped around the histone octamer and clipped by the H1 histone is known as nucleosome, right? This structure is called a nucleosome. Correct, everyone? And this structure of DNA, so multiple nucleosomes present together, and this packaging is known as beads on a string structure. So it looks something like this. So there is DNA followed by nucleosome, DNA, nucleosome, DNA, nucleosome. So this free DNA in between is called a linker DNA that links two nucleosomes together. And on one nucleosome, there is around 200 base pair of DNA wrapped. Okay. And this is the how the packaging is done, the levels of packaging. So we start with the naked DNA, then they form nucleosome, then they are wrapped around the histone proteins. Then these nucleosomes formed chromatin, which is the bead on string structure. Then the chromatins condensed further to make chromatin fiber. And this chromatin fiber again packs further to give rise to supercoiled structure, which gives rise to chromatid and chromosomes. Okay. Right. And what was euchromatin and heterochromatin? We also have Reem joining. Hello, Reem. How are you?
I'm good, sir.
Yes. Reem, would you like to tell me what is a euchromatin and heterochromatin? Remember from last class?
Euchromatic, they are loosely packed and they are light stained.
Yeah, they are loosely packed. That's why they are light stained. So they do not retain the stain. Remember, I gave you the analogy of walking through a dense forest versus a light.
Yeah.
Yeah. Please tell me.
The heterochromatids, they are densely packed. That's why they're dark stained.
And what about the transcriptional status of these chromatins?
Euchromatic, they are active.
They are transcriptionally active. So transcription
they're
happens.
transcriptionally active.
And
And
heterochromatids,
heterochromatids, they are transcriptionally inactive.
Right. Good. Okay, coming to Wafia. Wafia, are you there?
Yes, sir.
Yes. So tell me about, take us through the first experiment that we discussed, experiments to search for the genetic material, the Griffith's transforming principle. What was that?
So he used the bacteria to do the experiment. Called bacteria's name is Streptococcus pneumonia.
Yes.
So he took two strain that is smooth strain and rough strain. And this smooth strain, they produce these tiny colonies on a cultured plate, and they are lethal strains. And strain 2, that is rough strain or arch strain, they produce rough colonies on a cultured plate and non-lethal strain. And through the experiment, Griffith concluded that arch strain bacteria was somehow transformed by the heat kill S strain bacteria, and this must be due to transfer of genetic material, but the biochemical nature of the genetic material was not defined from this experiment. So he achieved that it can be transferred from one
organism to another.
One organism.
Yes. So basically what he figured out was that why this S strain is called smooth strain, because S strain, write down, just add things in this experiment. The S strain synthesizes a smooth polysaccharide chain, coat, a smooth polysaccharide coat around itself, which provides virulency. Virulency is lethality, where the bacteria can now produce toxins and kill the host, which it infects. So this smooth polysaccharide coat provides virulency. Okay, this was known. So what he proved through this experiment is that some transforming principle, which he said is genetic material, enabled the R strain to synthesize the smooth polysaccharide chain, polysaccharide coat, and become the S strain. And S strain is virulent strain, which kills. Is it clear, everyone?
So.
Yeah. Now, as Wafia correctly said, that through this experiment, it was not proved that whether that genetic material is a DNA, a protein, a lipid, or what is the chemical nature of that genetic material. It just proved that there is something present in the cell probably present in the nucleus, which gets transferred from one organism to another and can cause change in the physicality, in the physiology, or in the physical appearance or abilities. Now, next from here, the curiosity in the scientific community was to figure out the biochemical nature. Okay, so there were experiments done to figure out what is the biochemical nature. It's called biochemical characterization of transforming principle. Okay. Now remember, here there are three scientists who tried to figure out the biochemical nature using the same experiment which Griffith did, the transforming principle experiment, using the same bacteria. Now, let me ask you this question. I ask this question in every batch, and this is for you to understand the logic. So this question is for Reem, okay? First, if you know that there are 3, 4, or 5 ingredients, okay, in something, and one of them is responsible for some allergy, that, so suppose you are eating biryani, okay, so it has multiple ingredients, right? When you eat it, you get allergic reactions. Now you may be allergic to any one thing present in it. How will you figure out which component you are allergic to? What is the logical way to do that?
I guess you go to the doctor.
There's no doctor available. Then you cannot figure out. We cannot take the bacteria to the doctor, no? I took the bacteria to the doctor and asked the doctor that which is the genetic material, tell me. The doctor's like, you are a scientist, I am not a scientist. So think like a scientist. How will you figure out that out of 10 components which are mixed together, so inside the cell there are proteins?
You taste all of them again separately.
Again, exactly. You purify them separately and you test them separately for that same allergic reaction to happen or not, right?
Yeah.
See, you're a scientist already. And this is exactly what these three scientists did. So there were 3 people whose name was, it's called Avery, MacLeod and McCarthy experiment. So their name was Oswald Avery and Colin MacLeod. I think it's MacLeod if I'm not wrong. MAC, and McCarthy. In this you have MC. So Avery, MacLeod and McCarthy experiment. That is a follow-up of the Griffith's transforming principle experiment. In this, they did exactly what Reem has suggested to do. So inside a cell, there was a lot of debate going on at that time when people were trying to figure out what is the genetic material. And since we know that inside the cell, the most important roles, the most important functions are played by proteins, right? That's why we call protein as the building block of the cell itself. Yes or no? So people thought that protein could be the one thing which is doing all of this. Okay, so there were few candidates, for example, lipid molecules, because the cell membrane is made up of lipid. So lipid is also synthesized from Golgi bodies. Then it could be protein, or it can be nucleic acid, RNA or DNA. So these 3 were the potential candidates to be the genetic material. Now it was not sure which one is the genetic material. So Avery, MacLeod and McCarthy, what they did, they purified. So write down first point. They purified the major biochemicals from the cell. Purified the major biochemicals from the cell. And what were these major biochemicals? The targets: proteins, lipids, your RNA, DNA, etc. Okay, they separated all of it. Then what is the next step? What will you like to do, Reem? They separated. Now
They taste it.
what will happen if they will taste it?
They'll get some kind of idea of what caused
What?
the allergic reaction.
But in Griffith's transforming principle experiment, there was no allergic reaction. This is the experiment. R strain bacteria got converted into a S strain bacteria. So what will happen if they taste a lipid or a RNA or a DNA? They would not like to taste something taken out from the bacteria, right?
They will inject it.
Exactly. Where they will inject it?
The mice.
But if they will inject it in the mice, then the mice may not die, no, because RNA is transforming the R strain. It is not responsible for killing the mice directly. So what is happening is R strain is getting transformed into S strain due to something, right?
Yeah.
And then
They will separately inject it.
they will see that which of these purified biochemicals can transform the R strain into S strain. Okay. So what they will do is they will grow the R strain bacteria in the presence of just the proteins taken out from the S strain. So they purified the biochemicals from the cell of S strain bacteria. You understand? Then next point, write down. They checked which one could transform R strain into S strain. Is it clear? You cannot inject things in the bacteria. You can just culture the bacteria in the presence of it. Okay, at that point, at that time. So if you one by one you allow the R strain to take up protein, lipids, RNA, and DNA, and they figured out, write down, they discovered that only DNA was able to transform only DNA from the S strain. Let's write it. Only DNA from the S strain was able to transform the R strain into S strain. Is it clear? Do you think it is proof enough to say that DNA is the genetic material?
Yeah, sir.
Yeah, I also think so. But the way these experiments were conducted, and they also, like as a result, so what I'm writing in green are the results. They also, to cross check, they used enzymes. So they treated the cells with protein digesting enzymes, lipid digesting enzymes called proteases, lipases, and nucleic acid digesting enzymes called nucleases. So you have studied in class 11th in digestive system chapter. In our body, we have different enzymes: proteases, lipases, amylases, and nucleases to break down certain things, right? To digest certain molecules, correct? Yes or no?
Yes, sir.
So if you use proteases, then it will degrade all the protein that you have added from the S strain. Okay. And if you digest the component which was responsible for transforming, which in this case is the DNA. So only if you add DNAs, the transformation will be affected. So the transformation which should happen will now not happen. So write down. They also discovered that protein digesting enzymes called proteases, lipid digesting enzymes called lipases, and RNA digesting enzymes called RNases did not affect the transformation. Because they were not responsible for transformation. But what will affect the transformation? DNases. Okay. Only DNA digesting enzymes called DNases inhibited the transformation. Is it clear?
Yes,
So
sir.
this is just a proof of principle. It's just to cross check that yes, DNA is the genetic material. So they concluded, I write it here. They concluded that DNA is the genetic material from their side. So are you convinced, all of you?
I am, sir.
Yes.
Yes, sir.
I also
Yeah.
think that it is convincing. It's enough. It's a convincing enough proof. But not all biologists were convinced, because the way these experiments were done, if you are treating with something and you are growing the bacteria in the presence of something, all biologists were not um, actually convinced that it is a good way to do the experiment. Now when we read it in theory and we realize that when you just put DNA, the transformation happened. When you put other things, transformation did not happen. But there were other factors also that could affect it. So when you carefully look at the experimental design, there were some loopholes, and all scientists were not convinced at that time. So this was a proof closer to figuring out that DNA is the genetic material, but it was not a unequivocal proof. Like it was not a guaranteed proof. Okay. So every MacLeod and McCarthy did a brilliant experiment, brought us closer to finding the genetic material, but not everyone was convinced. So something else, there has to be some other type of experiment done which is more conclusive. So in science, the reputation of the scientists, or just because someone is a senior or a junior scientist, or someone is a teacher or a professor, you do not just agree to what someone who's with experience telling it. You have to trust the logic and the way the experiment has been conducted. If there is any any loophole, then that experiment will not be accepted as a concrete proof unless and until you plug all the loopholes and make it foolproof experiment. So that's where there were two people who gave the unequivocal proof, and it is known as the the Hershey-Chase experiment. Okay, so now I go to third thing here. Write down. The next step was the Hershey-Chase experiment. So just remember it like a story. Someone told something, then there were people who went on one step forward to tell something else, and it kept moving. So science always progresses through multiple scientists. So Hershey-Chase experiment comes from the name. The one scientist was Alfred Hershey. And the other was Martha Chase. Okay. They did this in 1952. And this experiment is regarded as, this experiment is regarded as the unequivocal proof that DNA is indeed the genetic material. Now, to prove it more clearly, you cannot take another biochemical approach, because they took the biochemical approach already. They already purified the biochemicals from the cell, Avery, MacLeod, and McCarthy. They did all which could have been done logically, like put one by one and see the transformation if it happens or not. But because of the experimental design, everyone was not convinced. So you had to take some other approach now, right? So what could be that other approach? So they come up with a very brilliant idea. So you know about viruses, everyone? Viruses? What are viruses? Madhya?
Viruses actually are non-living in the environment, but once they enter any body, they live the way that body is functioning.
Yes, and what is the, how do the viruses, how are they structured? They do not have a cytoplasm and other cell organelles, right?
Yes, they do not have a cytoplasm.
What do they have? They just have.
They just have DNA.
Just DNA in air.
In the nucleoplasm.
They do not have a nucleoplasm. They do not have any plasm. They just have a protein coat, and inside the protein coat there is a genetic material, or the DNA packed. Simple, right?
So they also have RNA, right?
They can have RNA. There are viruses which have, so they do not have both. They either have RNA as the genetic material or DNA as the genetic material. Okay. You understand? It's Mehraj was unmuted by mistake. Okay. Okay, sorry for that. Yes, Reem, what were you saying? They also have RNA, correct?
Yeah.
So, but at the same time, they do not have both RNA and DNA, right? Either they have RNA as the genetic material, so they are called RNA viruses, or they have DNA as the genetic material and they are called DNA viruses, right? But in case of both RNA and DNA, if you just prove with some other method that yes, DNA is actually the thing which is getting transferred, and if you can visualize it, if you can see it, then it will be a unequivocal proof, right? We can see it with our eye that what is getting transferred is DNA. And taking virus is the best system because viruses just have two things, a protein coat and a DNA inside. Okay, so for this they took a bacteriophage. You know what is a bacteriophage? So the model organism they used were bacteriophage. Now the name has bacteria in it, but they are not bacteria. Bacteriophage are the viruses that kill bacteria. So they only attack bacteria. Okay, viruses have this very interesting. So the one of the characteristics of viruses is that they are very specific, host specific. So if bacteriophages attacks the bacteria and kill the bacteria, they will not affect any other organism. Okay, they will only affect bacteria. So they are specific. So there are specific bacteria viruses, there are specific plant viruses, there are specific human viruses. Okay. Now bacteriophage has this structure like this. So they have a protein coat. Okay. With the neck region. And towards the base, they have these leg-like structures. Sit on a bacteria. You understood? So this is the protein coat, everyone. Everyone understands? And inside, there will be a single DNA. Okay. So everything, this neck region, all is made up of protein. Only what is in black is in protein and what is in blue is the DNA. So viruses are very simple. They have a protein and they have DNA. Now they took advantage of this system, this very simple system, and they did this. So write down what they did. Write down first what is bacteriophage. So write down. Bacteriophages are viruses that infect bacteria. Infect and kill bacteria. Bacteriophages are viruses that infect and kill bacteria. Okay. Next point. A bacteriophage, how it infects. Okay. A bacteriophage attaches itself to the bacteria. A bacteriophage attaches itself to the bacteria and sends its genetic material inside the cell, bacterial cell, and sends its genetic material inside the bacterial cell. Okay. This genetic material produces more viral particles inside the bacteria. This genetic material produces more viral particles. So because viral genetic material will have information about the viral particle, right? So there are many structures in this virus which will be formed separately and then assembled to make this virus. You understood, everyone? For example, this head coat, this neck region, these leg-like structures, they all will be formed and then again packed in the form of a virus. And this virus will now burst open the bacterial cell, kill this cell basically, and will come out to infect other thousands and lakhs of other bacterial cells. You understand, everyone? This is how the infection happens. Yeah. So next point, write down. What they did was they grew some viruses. They grew some viruses on a medium that contained. They grew some viruses on a medium that contained radioactive sulfur. So there were two mediums. Medium 1 that contained radioactive sulfur. Now, what is the advantage of this? The idea behind this is, if you can separately mark protein and DNA and then visualize that which one gets inside the bacteria, then we will be able to say that whether a protein is the genetic material, which people used to believe in that time, because protein does all the important business in the cell. So they thought that protein might be the genetic material. But if protein goes inside, we will be able to see it because we will radioactively mark it. If DNA will go inside the cell, we will be able to see that also because we will radioactively mark DNA. Now they thought how to mark both of them separately. So they took the advantage of these components, sulfur, and so radioactive sulfur marks the protein. It labels protein. Why it labels protein? If you culture the virus. So if you culture it in medium 1, which contains radioactive sulfur, protein contains sulfur, okay? Sulfur is not present in DNA. So if you are culturing an organism in an environment, and what do we mean by radioactive? Have you studied radioactive isotopes in chemistry yet? Some isotopes actually are radioactive, so they can show, you can visualize this radioactivity with the help of your naked eye. You understand? Have you studied radioactive isotopes? That will make it easier, or no? Yes or no?
Yes, sir, we have.
Yes, you have. So you know that radioactive, so radium, which are there in your watches, it's radioactive. That's where sometimes you can see in the dark. Can you see the time in the dark? So something that glows in the watch. Earlier, analog watches used to have their hands marked with this radium. That was a radioactive element, so it glows. Okay, so if you allow the virus to just grow in a radioactive sulfur medium, over the time, as the virus will divide and make more copies, it will make protein also. And from the environment, the only sulfur which is available is radioactive. So all the proteins will start incorporating radioactive sulfur. So over time, this protein coat, the whole coat, will be radioactively labeled if you grow it in medium 1. You understand? If you grow it in medium 2, which contains radioactive phosphorus. You remember that phosphorus is a part of DNA? From previous class, yes, in the last class, yesterday I told you the structure of DNA. Remember that it has a phosphate group?
Yes,
E?
sir.
Yes. So phosphorus is only present in the DNA. It's not present in the protein.
At the starting.
Exactly, at the very beginning. There is a phosphate, then there is a sugar. Phosphate and sugar makes the backbone, and then there is a base which comes towards the inside. So that phosphate group gets labeled with radioactive phosphorus. So it only labels the DNA and it does not labels the protein. So these two medium can specifically either label the protein or label the DNA. So both of these are labeled. You understood? So now there will be 2 batches, and let me make the 2 batches like this. So in first case, which is medium 1, you will see that the labeling I'm doing is in red. Okay, so you will see that the whole virus is actually labeled. The protein code is actually labeled. Is it clear? But the DNA inside is unlabeled, right? So radioactive labeled protein coat and unlabeled or non-labeled DNA. Is it clear? In case of medium
Yes, sir.
2, what will happen? Now, the protein coat will not be labeled. Understood? So you cannot see it. It will not show you radioactivity. But the DNA inside will be labeled now. You understand? Unlabeled protein coat, but labeled DNA. Okay. Radioactive phosphorus. So over the time, when new DNA will be formed, all the DNA will contain phosphorus. And for multiple generations, if you do this culturing, you will get two kind of viruses, two kind of bacteriophages. One with labeled protein coat due to sulfur, and one with labeled DNA due to radioactive phosphorus. Is this clear? So we are ready to do the experiment now. Okay, everyone. So now the first step of the experiment is infection. So now you allow these two batches to infect a E. coli. So there is an E. coli. Let's say there is a bacteria on both the sides. Okay. Bacteria has double cell membrane, the cell wall. So let me do this. I hope you all are drawing with me. People, are you drawing it?
Yes, sir.
So in this case, you will see that this virus will come and sit, right? And it has DNA inside. In this case, we'll see this virus come and attaches itself. So first, it attaches itself to the bacteria, and then through this, it sends its genetic material inside. And how does it happens? It exactly happens like a drill. So these viruses have certain proteins which actually physically make a hole. They drill in the bacterial cell wall and the cell membrane to inject its genetic material inside. Do you understand? Now, if the genetic material is protein, so in this first medium, in this first case, all the protein is labeled, right? So if inside there is any protein which is acting as a genetic material, it will get injected inside. Yes or no?
Yes, sir.
In the other case, if it is DNA, which is the genetic material, the DNA will get injected inside. And after the infection, the second stage is blending. Now, what we will do is this bacteria, E. coli, will be agitated in a blender. Now, in a blender, you just agitate it, like shake it well, agitate it so that all the bacteriophages that were attached to the surface now gets detached. So this bacteriophage which was attached here gets detached, right, and goes away. Okay. In this case, this is the bacteria which gets detached from the surface. So blending is done so that the whole bacteriophage, the virus should not come down with the bacteria. Otherwise, we will get a wrong signal. So we just want this bacteria to inject its genetic material. We just want this virus to inject its genetic material inside the bacteria and then go away. Do you understand, everyone? Is it clear?
Yes,
Now,
sir.
in this case, what is left inside is the DNA, the genetic material, which is not labeled. In this case, what will be left inside will be labeled, right? And then the empty capsule will just be agitated and removed from the bacterial surface. Now, so this. And the third thing that happens, then we will centrifugation. What is centrifugation? We spin this bacterial column in a test tube. We will spin it at very high speed. So what will happen? Which is heavier, a bacteria or a virus? What is bigger, a bacteria or a virus? Can you see from here? If a virus has to affect a bacteria, can it be bigger than bacteria? If a virus has to infect a bacteria, can it be bigger than a bacteria?
Yes.
No. How can it be bigger than the bacteria? Then how will it affect the bacteria? It has to be smaller than bacteria so that multiple virus can attach itself to the surface. Can you ride a mouse? Can you attach yourself to the surface of a mouse?
No.
You can do it to the surface of an elephant, right? But not a mouse. You understand? Can you be a pathogen or can you be a parasite to a mouse? No. Parasites, pathogens, those who infect are always smaller than the organism. Yes or no? If the organism which is killing the other organism is bigger, then we do not call it infection. We call it predation. Like a lion can actually kill a deer or a baby of the deer, right? That is called predatory behavior, predation. We hunt, we kill, that's different. Here it is an infection. So everything that infects us goes inside our cell and makes it copies. So I told you that these viruses, the genetic material go inside and then make multiple viruses inside. So this is a hint that they are smaller, correct? If they were bigger, how can they make their copies inside the cell of a bacteria? Yes or no?
Yes.
Yeah. So bacteria being bigger will always settle at the bottom. So when you will centrifuge, upon centrifugation, you will see that all the bacterial cells, the E. coli cells, will settle at the bottom, right? And the viruses will present in the supernatant. So the fluid in which small viruses will be present. So this is called supernatant, where the viruses, bacteriophages will be present. And this is called the sediment, where the bacterial cells will be present. Because they are heavier, so they will settle down. Do you understand?
Yes, sir.
Yes. And now in case 1, which was labeled? The protein coat or the DNA? Which was labeled in medium 1? Which was radioactively labeled? Protein coat. And that protein coat was then detached in the second step called blending, right? So you will see signal, radioactive signal in the supernatant, because all the protein coat is in the supernatant. You will not see any signal, any radioactivity in the cells. So this proves, so write down radioactive signal in the supernatant. And what are present in the supernatant? Sulfur 35, which is marking the protein. So which means that protein does not go into the bacterial cells as a genetic material. Do you understand? And in the other case, there will be cells sedimented at the bottom. And again, there will be a supernatant where the protein coats of the bacteria will be. Now, what is labeled here? Protein or the DNA?
DNA.
So where will you find the radioactivity signal? In the cells. Right? Yes or no?
Yes.
Yes, sir.
So the radioactive signal will come from the bacterial cells. Detected in the bacterial cell. And what we used here? Radioactive phosphorus P32, which mark the DNA. Okay. So this is a proof, and it is an unequivocal proof that we see with our eyes that when DNA was passed in, when we marked the DNA, okay, when we labeled the DNA versus when we labeled the protein, we saw that what goes inside the bacterial cell is the DNA. So we get the radioactivity signal in the DNA labeled medium from the cells, whereas we get the radioactive signal from the protein labeled medium in the supernatant. Do you understand? Everyone?
Yes, sir.
Yes. So write down. Bacteria results, conclusions. Write down the first, draw this. Everyone has drawn this? Let me know when you're done. Let me know when you're done, people. Then I'll move forward.
Done, sir.
Done? Everyone?
Yes, sir.
Okay. So write down conclusions, the results. Bacteria which was infected. Bacteria which was infected with viruses having radioactive DNA. Bacteria which were infected with viruses having radioactive DNA were found to be radioactive. Were found to be radioactive. Whereas bacteria which was infected with viruses having radioactive protein, whereas bacteria which was infected with viruses having radioactive protein were not radioactive. This proved that DNA is the genetic material that is passed on from viruses to the bacteria during an infection. This proves that DNA is the genetic material which is passed on from viruses to bacteria during infection. Is it clear, everyone? Written?
Yes, sir.
Great. Now, the question now is that you proved through bacteriophages, because bacteriophages have DNA as their genetic material. But what about RNA? So we now got to know that DNA is indeed the genetic material. But there are two different kinds of nucleic acids. One is DNA, deoxyribonucleic acid, and the other is RNA, ribonucleic acid, right? So can both be the genetic material, because both contains phosphorus, right? One is single stranded, one is double stranded, but both have phosphorus, sugar, and the bases, right? The components are same, yes or no? Yes?
Yes, sir.
So both can be the genetic material, right? Both has to be the genetic material. And it's true that both are genetic material. But here the debate started that which is a better genetic material and which was the first genetic material. So the properties of genetic material were then figured out and studied. Now, if I ask you, like if you have to choose a molecule, let's say, if I ask you logically, what should be the properties of a genetic material? If something has to act as a genetic material in our cells, what should be the property of it? What will be the ideal genetic material be like chemically? Any idea?
Passing down the genes to further generations.
Yes, correct. So it should be able to, what do you mean to say is that it should be able to express itself in the next generation, right?
Yeah.
Yes, right? Yeah, correct.
Yeah.
Very good. That is one criteria that genetic materials should have, that they should be able to express themselves in the next generation and they should be able to pass on to the next generation. Basically, you mean to say that they should follow Mendelian inheritance. Yes? What Mendel found in the pea plant, this genetic material should follow that inheritance pattern, right? Great. But apart from inheritance, any other property that you think should be there in a genetic material? What should be the characteristics of a class monitor or a team leader? You know, we look at some certain characteristics. Like he should be extrovert, that person should be good with people, good with numbers, you know, should be good with communication skills, leadership skills, right? Good speaking skills, yes or no?
Yeah.
So there are certain characteristics for certain roles that they have to play. So what should be the characteristics for a genetic material? It cannot be any random molecule in the cell which you can make a genetic material. So one, you told very correctly that it should be able to inherit from one generation to another, and it should be able to express itself in the next generation following the laws of Mendel, following the Mendelian inheritance. But before that, don't you think that it should be self-replicating? It should be able to replicate itself when the cell is dividing.
Yeah, it should.
Yes. So
Yeah.
the first and the foremost property of a genetic material, because we know that every cell contains genetic material, and every cell's genetic material then can give rise to other type of molecules. So all the information is coded. The recipe for every dish which is there inside the cell is written in the genetic code, right? So the genetic material should be able to write down. It should be self-replicating, which means generate a copy of itself. This is how it will be able to continue existing from one generation to another. Am I correct? What else? What else do you think should be the properties of a genetic material? See, I'm not here making you memorize things. I'm making you think and figure out for yourself. If you know, if you can think in the class and give me the answer, you do not need to remember this answer anymore because you thought it and it was a correct logical thinking. So all you have to do is to, just go and write your logic in the exams. So if I ask you today, what kind of a person should be the captain of a cricket team, you will tell me things, right? The person should be good with leadership qualities, should have served, should have experience handling a team, playing with a team, should be knowing each and every player's strength and weaknesses, when to use which player. Yes or no? So do you sit back and learn all these points? You just know it by your logic. Yes or no?
Yeah.
The same thing you have to do for science, for biology. So it should be self-replicating, makes sense. Second thing, if it is going to be the genetic material carrying the most important vital information in the body, in each and every cell, it should be stable, right? It should not be very reactive. Yes or no?
Yeah. Yes.
It should be both chemically and structurally stable. Correct? Then only it can continue to live as long as the organism lives for. So what is the average lifespan of humans? How long do humans live?
8 years.
78 years, right?
Yeah.
And they say that cells keep on dying and new cells keep on forming. That's true. But even when they are dying and forming, they have a life of their own. So every cell has a DNA that lives for very long, right? So it should not change in between. It should not be unstable, because if it gets unstable in between, then the moment the person reaches the midlife, the DNA or the genetic material will be unstable and degrade. That should not happen, because we need that genetic material throughout our life. So it should be chemically and structurally stable. What else? What else? As we live, as we grow, as species live, do they also evolve? Do they also change? The kind of human species that used to live, let's say 1 lakh years ago or 50,000 years ago, are we same like them?
No.
No, we have changed a lot, and the coming generations will change a lot. Earlier our ancestors, who were not Homo sapiens but other species in the genus Homo, or even the sapiens, have changed a lot. Our ancestors were living in wild, they were more hairy, they have they were less intelligent than us, because they were more what you can say, fierce with wild instincts, because they were living in wild, constant risk of dying or being predated. So they were predators and hunters. We are not predators and hunters. We are farmers, writers, artists, scientists. So we have grown we have evolved more intellectually, right, than physically. You understand? So all these changes are also happening through the genetic material, through the genes. We are incorporating some good things in the genes, and we are discarding the things that we no longer need. Like now, humans do not have hair all through their body, right? There are only certain restricted regions where we get the hair growth. We do not get a very lavish, thick hair growth throughout the body, but our ancestors used to get that. You understand? So the genetic material should also provide the scope for slow changes like mutations to bring in genetic variation. You understand, everyone?
Yes, sir.
Suppose you go and start living in a very hot country like African country, your skin will start getting tanned, right? There will be changes happening in your genetic material over the generation that will help you and your future progenies adapt well to that environment. So write down, third quality is it should be flexible for slow changes, for incorporating slow changes like mutation. Okay, that is required for better evolution. Excuse me. Okay, that is required for better evolution. And then coming to the point that Reem pointed out in the first place, that it should be able to inherit itself, right?
Yes, sir.
It should be able to express itself in the form of Mendelian characters. Right? These should be the ideal characteristics of a properties of a genetic material. Now, please write it down and think from the perspective that we are going to discuss next. Think all of these things from the perspective of a DNA versus and RNA. Okay, write it, take 2 minutes. I'll just grab myself water and I'll be back. Okay, and think about DNA and RNA, which fits better for all these properties. Okay, I'll be back in a minute. Okay, Reem?
Okay, sir.
Okay. Everyone is there? People, I'm back. Am I audible?
Yes, sir.
Yep. Okay. So, did you think about these properties from the perspective of a DNA and a RNA? So, let's go to the first one. It should be self-replicating. Now, remember I told you that at one point of time, people were thinking that protein could be the genetic material when it was not found. Before, when the Griffith experiment was done, they thought that it could either be protein, right? But can protein self-replicate itself? Just give me one moment.
No, sir.
I need to plug in my laptop. Just one. Yes, continue the discussion. Can protein self-replicate itself? How are proteins formed in the cell? Yes, Reem. Reem, how are proteins formed? Class 10th, sorry, class 9th. Cell, the fundamental unit of life. You read about a organelle called ribosome. Remember? Some memory refreshment?
Yeah. Yeah.
Yeah.
They
What?
form in the ribosome.
Not in the ribosome, through the ribosome. Ribosomes produce proteins, right? By reading the mRNA. Correct?
Yeah.
Yes?
Yeah.
So proteins have to depend on ribosomes and mRNA for its synthesis. Yes or no? So protein, if you think that protein could be a genetic material, it fails right there at the first step. Yes or no? It is not self-replicating. It cannot replicate itself without the help of other substances in the cell. Yes or no?
Yes, sir.
So protein can not be the genetic material for sure. That's done. Okay, very well. I have plugged in. Yeah, okay. So protein fails right here at the first step. So that's where we no longer believed, or scientists no longer believe, that it could be protein. And anyways, Hershey and Chase has given the unequivocal proof that it's not protein, it's the DNA. But now the debate is between DNA and the RNA, which is better as a genetic material. So both are self-replicating. That's okay. Which is chemically and structurally more stable? What do you think? Which is more stable? DNA or RNA? Any idea? First thing is stability. Which one do you think is more stable?
The
DNA.
DNA,
Why?
because
think
it
of
is a double helical structure.
it first. Yes, you're right. First, it is double stranded. Okay, so it does not have open ends. Second, so write down an RNA is more reactive and easily degradable. RNA is more reactive as compared to DNA. As compared to DNA, and easily degradable due to the presence of due to the presence of two prime hydroxyl group due to the presence of 2' hydroxyl group in every nucleotide, in every RNA nucleotide. Is it clear, everyone? So this is an open group present in every RNA because the RNA is a single-stranded. To make a double-stranded bond, this group has to bond. So when it is not, it is a single strand, then these groups are open, they are present. Okay, so this makes it more reactive. This group can react with other substances and makes it easily degradable. Is it clear? And on the other hand, right, for DNA, am I audible, people?
Yes, sir.
Let's write down. DNA is less reactive, chemically less reactive, and structurally more stable. DNA is chemically less reactive and structurally more stable. So on the basis of stability, which among the two do you think is a more suitable genetic material? DNA, right? So write down. Among the two, nucleic acids. So both are nucleic acids, right? Among the two nucleic acids, the DNA is a better genetic material. That's why more evolved organisms have DNA as their genetic material. Okay, everyone? All animals, all plants, they have DNA as their genetic material because it is a better genetic material. Viruses only have RNA as their genetic material. So this is on the basis of stability. Okay. Now let's talk about changing or evolution or evolving capability. Which one do you think will evolve faster or change faster? DNA or RNA? Wafia, Madhya, what do you think? Which will mutate or change faster?
RNA?
Yes, why? What is your logic?
Because it degrades faster.
Then it should degrade, no? It should not change. Degradation means damaging, done, gone. So if it degrades faster, it will just finish. Evolution?
Because it is more reactive.
Yes, right. Because it is more reactive, so it will do more reactions. Most of the time, if it will not degrade, it will change, right? Write down RNA. So both DNA and RNA are able to mutate. Okay. So first write down this point. Both DNA and RNA can mutate. Both DNA and RNA can mutate, but RNA being more reactive, and less stable. Mutates at a faster rate. Okay, so that's why viruses that have RNA as their genetic material mutate and evolve faster than viruses or organisms who have DNA as their genetic material. You understand? Yes? That is why it is difficult to treat the infections which are caused by RNA viruses. Now you understand what is the reason behind this? Because by the time we try to track it down, we try to kill those viruses, they mutate and change into something else, like some other genotype. Making sense, everyone?
Yes.
So RNA being more reactive and less stable mutates at a faster rate. So RNA viruses have shorter lifespan and they evolve faster. So RNA viruses have shorter lifespans and they evolve faster. Okay. So in evolution, in evolving capability or in mutating capability, RNAs are better. They mutate faster. But mutation is not always better. No, it is not always good, right? So we realized that in some things DNA is better, like in stability, but in reactivity, RNA is better. So what can you summarize from this? Can we say that RNA is better for the transmission of genetic information? It can get transmitted better. Now, why I'm saying this is because if we see the central dogma of biology, right? What was the central dogma of biology? Again, tell me. Wafia. What is central dogma? The flow of genetic information. What was it? DNA to RNA and RNA to proteins. Correct? Yes or no? Wafia.
Yes. Yes, sir.
Yep. So DNA makes RNA, RNA codes for protein. So if we see it from here, RNA can directly make proteins, right? Whereas DNA has to depend on RNA to make proteins. Yes or no?
Yes.
And ultimately in the cell, in the living system, all the functions, all the genes are expressed in the form of proteins are the expressed products. So whatever has to be expressed in the form of protein. They are the functional units. So which one is better for transferring the information, RNA or DNA?
RNA?
Yes, RNA is better, right? So write down. RNA can directly code for synthesis of proteins. RNA can directly code for synthesis of proteins. Whereas DNA has to depend on the RNA to synthesize proteins, whereas DNA has to depend on RNA. For protein synthesis. Yes or no?
Yes, sir.
So all the protein synthesis machinery, in other words, if I say the protein synthesis machinery of all the cells, whether it is prokaryotic or it is eukaryotic, it is evolved around the RNA and not around the DNA, right? So RNA has to be there either as a mediator in between, or in some cases it could be the direct genetic material. So if I summarize in one line, then I can say that both DNA and RNA can act as genetic material, right? Yes. So this is the conclusion in one line. So both RNA and DNA can serve or can function as genetic material. Correct? But also what is true is that DNA is more stable, right? And is preferred for storage of information. So if you have to store the information, which is the genetic information in this case, then which one will you like to rely on, DNA or RNA, for the storing of the information? When you
DNA.
store, yes, when you store information, let's say in a pen drive or something, you depend on something which is more stable, right? Which cannot get corrupt easily. Yes or no? Which is safe for your information. So DNA is more stable and hence preferred for storage of information. But there is also one advantage with RNA. What is the advantage of RNA? RNA is better for transmission of the genetic information. Correct? Transmission to the next generation, right? So, or in the same cell, like making proteins. So RNA is better for transmission of genetic information, and DNA is better for storing the genetic information. You understood about DNA versus RNA? All the three things? Everyone understood?
Yes.
Yes, please note this down. Please complete this and let me know. Let me know when you're done, people. Done?
Yes, sir.
Others? Wafia? Done? Okay. Cool. So you have understood till here, no doubt, right? Now we understood that it is DNA, that is the genetic material. We also understood that DNA and RNA both have the potential to be genetic material. In fact, they are genetic material in different organisms. We also realized that DNA is more stable and hence is more preferred form of genetic material for storage of information. It is better in that way. But RNA is better for transmission of genetic material. Okay. Or transmission of genetic information, right? It's better that way because it's more reactive. Now, immediately, the next question that comes in our mind, I'm sure, is which was the first genetic material? Like, which evolved first as a genetic material? DNA or RNA? What do you think?
RNA.
RNA? And why do you think so? Was it Madhya?
Because
Yes,
RNA
Madhya.
could
Yes.
be formed easily from the proteins, and DNA is dependent on RNA for its formation.
RNA is not formed from the proteins. Protein is formed from the RNAs. Okay.
Yeah,
In fact, RNA is formed from the DNA. Remember? DNA makes RNA, RNA makes protein. So to make proteins, DNA is dependent on RNA. There has to be a RNA in between. But RNA either is made from the DNA or it can be self-formed, like it can form on its own in the cell. But focus on the characteristics. Okay, so you are giving the right answer, Madhya, that RNA is the first genetic material. There are evidences to support this hypothesis. So it is known as the RNA World hypothesis, which now has become the RNA World Theory, because there are evidences to support it. So you're right, but your reasons are not correct. So you said that, would you like to try it again? What are the properties of RNA that help it to become first genetic material over DNA?
It's self-replication, the property of self-replication.
Yes. So RNA can self-replicate. That's one. Second thing, RNA is more reactive, right? And apart from
Yes.
just acting as a genetic material, it can also carry on other reactions in the cell. Okay. And let me tell you, RNA can also act like an enzyme. Mostly in our bodies, enzymes are proteins, right? All our enzymes are proteins, lipases, proteases, all of them, they are proteins basically. But there are certain processes in our body as well, which are carried out only by RNA enzymes that are called ribozymes. So this is a proof that RNA is not only able to store in, like, act as a genetic material, transfer the genetic information, it is also able to carry out certain reactions in the cell. So it is multi-talented, it's multi-purpose. It has a multi-purpose use. You understood, everyone? Whereas DNA is more stable, more evolved, but it does not catalyze reactions. It cannot serve as any other purpose. It has a specialized purpose of being the genetic material. Now let me give you an analogy, okay, Madhya? So let's imagine that Madhya is starting a company. Just like a cell, the first living system is being formed. So if you are starting a new company or a new group, you know, and Wafia and me both comes to you, for a position. I say that you need a position for example, let's say you need a manager, and I say that I am very equipped and stable and specialist as a manager. I do the managing business very good, so hire me. Okay, but you're starting the company, so you're starting it, you have limited funds, you have to grow it, right? And Wafia comes and says that, I can manage things, I'm a good manager. Along with that, I can also handle your accounts because I also have some experience in accountancy. So Wafia is neither a specialist in managing nor a specialist in accounts, but she can do both the things fairly good. So who will you like to prefer to hire during the initial days of your company, Madhya?
Someone who could do both the things
Right?
fairly
You
good.
will hire, yes, you will hire Wafia because your first motive right now is to get the company up and running, to make it to a level where you can afford to now evolve and make certain departments, then put the head of department who are specialists. Yes or no?
Yes, sir.
Yeah, so that's the logical approach, and that's the logical approach for a living system as well. So when living systems were evolving, the first living system could not afford to, make 10 different specialists at every point. So that system will first go with something that will be multifactorial, that can even act as a genetic material, and when required can also catalyze some reactions in the cell so that the cell can keep going, can keep evolving. You understand? Over the time, when the right time will come, DNA will evolve from the RNA and take its place. Do you understand? Everyone, is it clear from the discussion?
Yes, sir.
Yes, sir.
Yes.
Yes. So write down RNA was the first genetic material. Let's write down as some points. RNA was the first genetic material. Many essential life processes, for example, metabolism, translation is protein synthesis, etc., has evolved around RNA. Okay, not around DNA. This is the second evidence. So the first evidence. Second evidence is RNA can act, or RNA can serve as a genetic material as well as a catalyst. Okay, and such RNAs are called ribozymes. An enzyme which is an RNA is called ribozyme. Okay, there are two words: ribonucleic acid, ribo, enzymes. So ribozyme is a ribonucleic acid which is an enzyme. Okay. Next point, there are important biochemical reactions in the living system that are still catalyzed, that are still catalyzed by some RNAs, and not protein enzymes. So this is a proof, right? If it is still doing that, it must have been doing that when the life was evolving very early on. Yes or no? Because it is present before protein came. Yes, everyone? You understand?
Yes, sir.
Yes. So these are all the the proofs that tell that RNA was the first genetic material, and from there onward, because RNA was less stable and more reactive, so because write down, due to RNA being less stable, over time, a better genetic material, better means more stable, a better genetic material, DNA, evolved from RNA. Okay. Is it clear, everyone?
Yes, sir.
So what did we learn today? We learned till the RNA world. We realized that RNA was the first genetic material, and this concept or this theory is known as the RNA world theory. At one point of time, the world was only of the RNA. The genetic material was only RNA. This is when the early life was beginning on the planet Earth. Okay. So from here onwards, we will now from the next class, I will start with the three processes of central dogma: replication, which is DNA from DNA; transcription, which is RNA from DNA; and translation, which is protein from RNA. So these three things I'll do in detail from the next class. Do you have any doubts still here about the DNA, RNA, anything, any experiment? Yes, no?
No, sir.
No doubts? Okay, so Madhya, please